little special episode for our Nobel predictions.
Yes, I'm very excited.
It's the biggest week of science of the year.
Yeah.
We're going to start with Monday, which is the medicine and physiology. So what are you thinking?
All right. So I think there's two possibilities. Okay. The first one I'm thinking goes to cGAS-STING, which is a signaling pathway that tries to catch rogue DNA molecules in our cells.
Right.
Okay. We're eukaryotes, right? And what that means is all of our DNA is packaged inside of the nucleus of every one of our cells. The genetic material that's on the outside of the nucleus is all in the form of RNA. Whenever there's a DNA molecule outside of the nucleus in the cytoplasm of the cell, it's a red flag. Okay.
An invader.
It's either an invader that's like a virus has come in, or the cell is losing it, right? And it's like a cancer cell. So either way, it's something that you're going to need to catch and then, figure it out. So that first part, catching that DNA molecule and then starting this signal transduction pathway, that is the cGAS-STING pathway.
Got it.
It's part of our innate immunity. So there's three researchers, Jijian James Chen at the University of Texas in Dallas at the medical center there.
Yep.
Then there's Glenn N. Barber. He's from the Ohio State
Ohio State.
And then finally, we've got Andrea Ablasser. She's going to be quite young if she wins. She's at the École Polytechnique Fédérale de Lausanne in Lausanne, Switzerland. The second one that I'm thinking is something called optogenetics. Okay, optogenetics is this technology that was developed to let us as researchers control cells using light. Okay, it's a combination of using light to basically tune this protein that can then turn on and off neurons. So what you're seeing over there are neurons that are expressing a specific kind of protein on their membrane. These are called channel rhodopsins. So they respond to light. And when they absorb a photon or two of this light, it actually changes their configuration. Because the protein literally changes its shape. You can open and close ion channels in the neurons, which is literally how you turn on and off neurons, right?
Right.
So you can on command be like, I want you guys to turn on now. Now you can do this in living animals.
Yes.
Right? You can have a little fiber optic cable that goes into their brain.
Yes.
And then as they're behaving, going through a maze, like a cheese maze or something, right?
Yes.
You can send in light at that frequency through this fiber optic cable.
So crazy.
And then in the middle of their task, as they're learning, you can be like, let's turn off this class of neurons. What happens? So there's three people. Peter Hegemann at the Humboldt University of Berlin. The second one I would say is Gero Miesenbach at Oxford. And then there's Karl Dieserath at Stanford.